Title of my talk is Elucidating the Role of Norepinephrine in White Matter Development and Disease. So just as a introduction, I'm gonna begin with a brief review of norepinephrine function in the central nervous system and what we currently understand the role of norepinephrine is in oligodendrocyte precursor cell function, or OPC function. And then I'll go over some of my data that I've acquired so far and what's ongoing. And then I'll summarize and talk about the many future directions that I have to go in. And then, of course, acknowledge all of the important people that have contributed to this. So norepinephrine, as many of you probably know, serves many functions in the CNS. The main hub of noradrenergic neurons in the CNS are located in the locus coeruleus in the pons. And these neurons project all throughout the brain in the neocortex, the hippocampus, the thalamus, the midbrain tectum, and even descending into the spinal cord to regulate pain. Norepinephrine is important in regulating mood, of wakefulness, arousal, alertness, and cognition. And in addition to that, it targets glia directly, which is what my talk is going to focus on, specifically in its role in targeting OPCs. So norepinephrine in OPCs specifically targets the alpha-1 receptor. It binds it directly, and there's data, very new data from 2023 is published out of 2 different labs showing that this binding increases calcium dynamics within the OPC. And what that means is still all being worked out, but certainly plays an important role. The norepinephrine also binds to other glial cells, beta receptors on radial glia in the hippocampus, on some astrocytes, and alpha-2 is primarily on the presynaptic side where we see reuptake of norepinephrine. The other protein enzyme I want to point out on the slide is DBH. I have here on the presynaptic side. It's dopamine beta hydroxylase is involved in converting dopamine to noradrenaline or norepinephrine. And this is important because it's specifically expressed in noradrenergic neurons, and that'll become important in my later experiments. So a little more on OPCs and why they're important. So OPCs, of course, are the precursors to myelinating oligodendrocytes and are present early on during embryonic development and are present in early postnatal and persist all the way through into adulthood. So here in this panel, I just wanted to introduce and show you that OPCs are important in hypoxic ischemic or HI injury. This image here comes from a paper published this year looking at HI injury in a rat model. Just for those not familiar with HI injury in these rodent models, usually what they do is a common carotid ligation on one side of the, on just one side, leaving the other side alone, and then they put the rodents into a hypoxic chamber for about an hour or so. And so then you can compare the ipsilateral side to the contralateral side in terms of the ischemic injury, and you can see that this is a stain for PLP, which marks myelination. You can see there's less myelination on the side with the ischemic injury. So OPCs are really important in HI injury. And this brings me to a hypothesis, which is twofold. So I'm looking at normal conditions. Under normal development physiologic conditions, I hypothesize that norepinephrine induces proliferation and differentiation in OPCs. There's some literature to support this, but no one's actually looked during development, only in adult animals. And then my second part of my hypothesis is that under extreme stress, like in perinatal hypoxic ischemia, there's abundance or a surge of norepinephrine, and this can lead to aberrant OPC proliferation, differentiation, and I'll be looking at myelination. So here's one of my early first experiments where I isolated OPCs from the mouse brain using a magnetic max cell sorting technique. And then in culture, I added norepinephrine at increasing concentrations to measure proliferation. So here you can see the cells that incorporate EDU are proliferating, and the OPCs are marked by PDGFR alpha and OLEG2. And at low concentrations of norepinephrine, I saw an increase in proliferation in this bar here. But as I increased the concentration of norepinephrine, I saw a dose-dependent effect and inhibition of the proliferation. To follow up this study, I added different norepinephrine antagonists to the cultures. I used that 1 micromolar of norepinephrine in which I saw the proliferation increase, and then I added varying concentrations of these different drugs. So I used an alpha-1 inhibitor, which is doxazosin, alpha-2, yohimbine, and then the beta antagonist propranolol. And you can see that the doxazosin successfully inhibited the growth of the OPCs exposed to norepinephrine, whereas the yohimbine and propranolol didn't have that same effect, which isn't entirely too surprising given that doxazosin is an alpha-1 inhibitor, and we know that OPCs are expressing the alpha-1 receptor. So this was a nice validation. So far I've shown you in vitro data, and the next part I want to talk about my current studies, which are in vivo and far more exciting. So this is in order to study the norepinephrine and noradrenergic firing within an in vivo model. I'm utilizing optogenetic stimulation of the locus coeruleus in neonatal mice. So optogenetics is a really eloquent tool in order that allows us to study specific neuron firing that's controlled by light. These cells express a photosensitive protein that allows us to, we can physiologically or like super physiologically control the firing of a particular subset of neurons. So in this model, I've crossed a DBH-CRE mice. DBH, remember, is that protein that's only expressed in noradrenergic neurons, with a carmine floxed mice, so that only noradrenergic neurons are responsive to far red light. And what I want to point out is what's really neat about this is that typically with optogenetics, we have to insert a fiber optic through the skull, which introduces a small injury, let the mice recover, but then you're able to specifically stimulate a particular region of the brain. I asked the question when I came to the lab, can we do this in pup mice without introducing injury? Because I don't want to introduce an injury when I want to study injury. With red light that penetrates deeper into the brain, and in pups that have very thin skulls without fur, can we just maybe stimulate these mice extracranially? So what's neat is it's taken several months, of course, to get these breedings done and set up this experiment, but I have some preliminary data showing that this is actually working. So here's some ongoing work, but you can see here that through extracranial stimulation, I'll start by just showing the DAPI, of course, marks nuclei, and then RFP marks all of those DBH positive neurons, and CFOS marks firing neurons. So we can quantify the number of noradrenergic neurons that are firing in response to the optogenetic stimulation. And so you can see I only have a couple of mice in each of these groups, so I'm still working, this is ongoing, but I can already see a significant difference between the mice that are DBH-CRE carmine. In other words, they're responding to the optogenetic stimulation. I'm seeing about 30% of those noradrenergic neurons are firing in response to light. And in my control group, which are the mice are undergoing all the same procedures, but I'm not turning on light, I'm seeing very few, about 1% firing. So again, this is ongoing work, lots of studies to be done still. To summarize, so far I've shown you that OPCs are directly regulated by norepinephrine, demonstrated by in vitro data, that we see an increase at low concentration of norepinephrine and an decrease in or inhibition of proliferation at higher concentrations. And that the noradrenergic system can be directly and specifically targeted in postnatal pups using a non-invasive extracranial optogenetic stimulation. Future directions, there are many, but I've sort of summarized some overarching ones for myself in this project. So my question is, how does the OPC response to norepinephrine impact development? And what role, if any, does this relationship play in perinatal injury like hypoxic ischemia? And I'm going to determine the effect of norepinephrine on OPC function and developmental myelination using this in vivo model under normal conditions, and then following HI injury, similar to the one I described earlier with common carotid ligation in hypoxic chamber. And lastly, I would like to utilize an ADRA1A conditional knockout. ADRA1A is is the gene responsible for alpha-1 receptor expression in OPCs. And so we'll be able to conditionally knock out alpha-1 receptors in OPCs and see how that impacts myelination in response to HI injury.
Hi, I'm Liz, and today I'm going to be talking about a project that I've been working on getting off the ground with the help of Dr. Vivek Butch and the members of his lab. And I'm talking to you today about network correlates of complex neurocognitive behavior in pediatric patients with intracortical EEG. And this is the button I push. Okay. So imagine you're a resident. So it'll be easier for some of us than for others. And you're you're getting ready to round on a busy day on the stroke service. You're responding to volts, you're putting in orders, you're getting ready to present your next patient. You've got a lot going on, but you're very engaged. And then Dr. Schwartz hits you with a question. What do you think the localization is for the stroke for this patient? Or what's the most relevant recent trial to dictate how we treat this patient? Some days, sometimes you might find that you fire back really quickly with this super thoughtful, comprehensive answer. And some other days, or maybe even later on the same day, maybe not so much, right? Maybe your response isn't so quick. Maybe it's not so comprehensive, maybe not so correct. And our lab is really interested in understanding, what are the factors that really kind of dictate those moment-to-moment fluctuations in your own cognition? You might think, on the same morning, certainly your own knowledge base is maybe not fluctuating that much, and certainly your own neuroanatomic substrate is not changing so much. So what are the network correlates? What's the network state that determines your ability for cognitive performance? And what's the network state that subserves better cognition? If we better understood that network, could we induce it in ourselves or in others? Or are there developmental time periods when that network state is particularly vulnerable to lesion or particularly amenable to intervention? Historically, research into cognition has been arguably a little bit piecemeal. It had almost like a lesional approach. And that's led to a lot of advancements in our understanding of networks of cognition. Specifically that the PFC is very important underlying attention, and that the hippocampus is very important in memory. But if you think back to the example I just gave you, from a practical perspective, cognitive performance really engages many aspects of the brain, right? And discussions of our capacity for memory are really almost nonsensical when uncoupled from discussions of our capacity from attention kind of at the same time. So clearly, cognitive performance is a network performance, a network process that requires engagement of multiple brain areas and multiple modalities. So here I'm showing diagrams of several of the networks that have been identified just in the resting state. And actually, multiple avenues of investigation have converged on the idea that aberrant network dynamics, whether that is decreased connectivity within these networks, or changes in firing frequency or changing in firing strength within specific spectral frequencies, are associated with worsened cognitive performance in aging, Alzheimer's disease, Down syndrome, and schizophrenia. But this still doesn't explain what I was talking about earlier, which is what determines fluctuation within one individual, within their own cognitive performance. And our lab is particularly interested in understanding an aspect of cognition called preparatory control. And this is the state whereby the brain is preparing to be is using prior information to prepare to be particularly sensitive to incoming goal-directed stimuli. So it's using, accessing different parts of the network to maintain relevant information in working memory and trigger goal representations, and coordinating your attention and your perception and your ability to react to a stimulus. And preparing to be better able to respond to a cognitive load. So the question that our lab is really interested in is, are there particularly network states that are primed to perform, that are primed to respond to a cognitive challenge? And members of our lab, of the Butch lab, have used stereo EEG to investigate network correlates of preparatory control during a cognitive task. So here I'm showing basically a schematic of an individual undergoing a stereo EEG for clinical implications. And while they're admitted for that clinical workup, we ask them to perform a computer-based cognitive task. And here I'm showing a very simple reaction time task. This is a temporal expectancy task that really assesses both kind kind of processing speed as well as kind of goal-directed attention. It's pretty straightforward. As I'm showing here, there's a visual fixation cue. And then as soon as that visual fixation cue changes color, we just ask the participant to press a button as fast as they can. And we're measuring their reaction time, and that's kind of our gauge of their cognitive performance on this very simple task. So while they're performing that task, we collect the stereo EEG, and then we're able to look at the correlates of their cognitive performance by first breaking this down into its constituent spectral frequencies of relevance, so alpha, beta, gamma, and high gamma, because this is stereo EEG, so it's intracortical. And from this, we're able to calculate phase locking between different electrodes or different nodes within the network. So basically, we're looking at the waveforms within each of these frequency bands and how locked are they, how in phase are they between different electrodes. And that gives us a measure of how interconnected is a particular node, a particular electrode within this network. What is the communicability of each particular electrode site, each particular node. And then once we have that measure of communicability or interconnectedness for each individual site within the network, we can see, with a regression analysis, how does that degree of interconnectedness correspond to cognitive performance. So here we're looking at reaction time. Faster reaction time is better cognitive performance. And so we're looking for a negative relationship. So are there points within the network where the interconnectedness or intercommunicability is predictive of faster performance? Or to the contrary, are there sites where interconnectedness is actually predictive of poorer performance? I'm showing results from some members of the lab. They didn't do it right. Their work on participants. They're not the participants. So here you can see that there are actually significant relationships within particular neuroanatomic nodes within the network that predict faster performance or favorable performance on this task. Those are shown in red. And I hope that you can see that primarily these are clustered within white matter anatomic nodes. So the thalamocortical, frontal association, and temporal association areas, whereas kind of interestingly, increased communicability or interconnectedness within the frontal gray matter is actually predictive of poorer performance or slower performance on this task. And here that's kind of just represented visually, showing the white matter areas that have been associated with more favorable performance, and then the gray matter areas that are shown with poorer performance. So overall, we can the pro-cognitive preparatory control network that is metastable. And by metastable, I mean consistent across participants. So stable across participants rather than just within an individual, is characterized by high communicability in the lower frequency bands, particularly in white matter nodes. But we wondered further, how does this translate into children? All of this data I've shown you so far is from work in adults. So how does this translate into children? What's the developmental time course? Are there critical periods during which this establishment of this network is particularly vulnerable to injury or amenable to intervention? This work is still somewhat in its infancy, pun intended. So here I'm showing you data from an individual patient, really kind of a proof of concept, who had implantation with 260 electrode contacts. So here you can see his implantation schema. And he was admitted for stereo EEG monitoring. Of course, we collected his data. And given the previous data that I've shown you, where it seems like the lower frequency bands are particularly relevant for cognitive performance, and focusing here on the beta spectral band, and looking at phase locking between each individual electrode point. So each individual electrode as compared to every other electrode, how in sync are they? So here you can see that on the x-axis, we have every single electrode contact from 1 to 260. And on the y-axis, we have, again, every single electrode contact from 1 to 260. The heat map is showing what is the phase locking value? So how interconnected is each individual electrode? And we're looking at slow trials versus fast trials. And I think that maybe you can observe here in this first, single participant kind of proof of concept experiment, we don't see any like really robust patterns kind of emerging. But one thing that I think is really interesting is when I looked at this data, I happened obviously I know this patient, and I knew that he had like a really robust, very active seizure network. That's why he's here, right? So I thought, well, okay, this is just his seizure network that we're looking at, and it's completely drowning out any kind of behavioral signal. So here, actually, I've highlighted the electrode from which all of his seizures originated and all of his interictal activity. And one thing I think is really interesting is you can see that it actually is really not driving this interconnectedness. It's not driving the phase locking value. So I think that's kind of encouraging that as we kind of pursue this further and expand this out to more children, we actually will hopefully be able to see the behavioral signal if it's there and how it evolves over time. So obviously we have a lot more work to do here and kind of expanding this out into more kids. But another kind of direction that we're really interested in going in the future is to ask the question, is the preparatory control network, is it consistent regardless of the cognitive domain engaged? Because as I've shown you, all of the tasks, all of the data we've shown so far has been from this temporal expectancy task, which you might argue is a quite simple task, and that the cognitive tasks that you engage in every day are a little bit more complex than that. So we wondered, is it the same preparatory control network that predicts favorable performance regardless of the cognitive load, its type, or its degree, which is somewhat of a crazy idea or a little bit heretical. If you think back to the beginning of my talk, talked about kind of the research into cognition thus far has really focused on, the role of individual brain areas in different cognitive domains. So to take that step back and take this approach and say, well, is there a single network that really predicts favorable cognition regardless of the domain that we're engaging, is a little bit of a new approach. So we've designed this pediatric cognitive battery, and really like the aspects that we focused on for this battery is that it would be brief, repeatable, and that it is kind of scalable across different ages and different developmental capacities and cognitive capacities, because obviously the population that is admitted for stereo EEG monitoring has really a broad range of cognitive abilities. And this battery is designed to test aspects of attention, processing speed, inhibitory function, and working memory. And so we'll use that to kind of investigate the preparatory control network and network predictors of favorable cognitive performance across domains. But unfortunately, this is still kind of a future direction. So there I will have to end rather abruptly and say thank you to all of my supporters, both within the lab and the department, and especially to Ben Sherman, who's pictured here as a particularly talented medical student who really is responsible for a lot of the legwork kind of getting this off the ground. And thank you for your attention.
Questions?
Hi, thanks, Liz. I'm curious, the loci that you saw in your network, is there a comparison to fMR data with bold signals? I know you're looking at deeper structures, but so my first question is there a parallel that you saw with the known networks found in fMR with these tasks? And the second thing is, in that literature, is there a difference between children in development versus adults? Because I think that's another thing you're trying to compare, right?
Yeah, really good question. So the unfortunate thing about fMRI is, particularly if looking at preparatory control, it really doesn't have the temporal resolution. So I think that's really kind of part of why the network correlates of this particular aspect of cognition in this task haven't really been investigated, because, like the peak bold signal can be up to seconds after presentation of a stimulus. And we're looking at microseconds before a response. So I don't know that there's necessarily like an fMRI correlate I can turn to. There have been some fMRI studies looking at cognitive networks comparing children to adults. And there is at least one study I showed that I'm familiar with that I think showed that the network responsible for cognition in that task, and I'm forgetting what the task was, but it seems to reach an adult-like state in the teenage years, which makes sense. And that prior to that, actually, it was an executive functioning task. Sorry, now I'm remembering. And that prior to that, the network is present but not as strong, which kind of makes sense. Like the connectivity measures are not as robust, and that seems to be associated with poorer performance on an executive function task. And then that seems to kind of stabilize in the teenage years.
Babak had a comment. He said, great talk. He wanted to know more about the state, like time of day or medications, noise, external noise? Like, how are you dealing with all those confounders?
Yeah, that's a good question. So, I mean, since we only have one patient so far,
But do you have a plan?
I think that one of the strengths of this approach, and that was used in the prior research as well, is so I have focused really here a lot in my presentation on the metastable networks, because I think the metastable networks, meaning those networks that are consistent across individual, because I think that's really exciting in terms of translatability. But really, obviously, the first layer of analysis for every single one of these is within patient. So within patient, identifying networks that significantly protect poor versus more favorable performance. And by using that kind of like within patient control, we're able to avoid a lot of kind of like the between patient confounders. And it also kind of allows us to take advantage of this clinically captured population where implantation sites are really diverse, right? So that's kind of another challenge in working in this population. By doing these within patient correlations first, we're able to kind of avoid those confounders. And in some ways, it's really pretty amazing that the prior work in the lab found anything metastable at all. If you think about all of those between subject factors, probably the most prominent of which, the diversity of implantation sites, the fact that they found anything at all is kind of incredible. So I think it's kind of those within patient, using the patient's own poor performance trials as the control for the favorable control performance trials that we're trying to avoid a lot of those confounders.
Very polished presentation. And you just alluded to what I was going to ask, which is that eventually you're going to want to generalize beyond the within patient to the across patient, especially if you want to be able to look over time and compare to adults. So without giving any equations, because it's too early in the morning for that, how does one try to approach that complexity?
Yeah, I think the developmental time course piece is going to be really challenging. And I'll be perfectly honest, I haven't had detailed conversations with any statisticians about how to approach that. I think it's going to be particularly challenging. So how we've approached it in the past in the adult data is really by trying to take kind of the neuroanatomical correlates and using I'm not familiar with this analysis because I have one patient so far, but basically by grouping electrode nodes that are like within, trying to make anatomical kind of subsets to group the electrode nodes. And that's one way that we kind of collapse the variable dimensions to do some kind of more meaningful comparisons. But I think that the developmental time course, to be perfectly honest, is not, I don't have a great statistical plan just yet.